Hello everyone. In this class, we'll try to learn about the functions of loop of Henle, distal converted tubule and collecting duct. Functions of loop of Henle. The loop of Henle has descending and ascending limbs. Loop of Henle of juxtamedullary nephrons are longer than the cortical nephrons and dip deep into the medullary pyramid. In juxtamedullary nephrons, descending limb of loop of Henle is totally thin, whereas ascending limb has thin and thick portions. All the segments, that is thin ascending, thin descending, thin ascending and thick ascending segments have different transport properties and permeability. Fluid from the proximal tubule that enters the loop of Henle is almost isotonic, whereas fluid leaving LOH is considerably hypotonic, having osmolality of 100 milliosmoles per kg of water in comparison to 8 to 85 milliosmoles of water at the proximal tubular end. That means it becomes hypotonic. This is because solids are reabsorbed in ascending limb of loop of Henle without reabsorption of water that makes the fluid more dilute. Reabsorption of solids into the interstitium from loop of Henle plays a critical role in the countercurrent mechanism of urine concentration. Cortical nephrons are more in number and loop of Henle of these nephrons help in urine formation, whereas juxtamedullary nephrons are less in number and loop of Henle of these nephrons help in urine concentration. Transport of solids here. Loop of Henle reabsorbs about 25% of the filtered sodium chloride, potassium, 30% of the filtered calcium, and 65% of the filtered magnesium. The descending limb of loop of Henle is permeable to water but not to the solids, whereas the ascending limb is impermeable to water. Sodium, potassium, chloride are co-transported out of the thick segments of the ascending limb. In the descending limb is permeable to water but not to the solids. Therefore, fluid in the terminal part of the descending limb becomes hypertonic as water moves out of the tubule to enter in the interstitial space. Thin ascending limb in the thin part of ascending limb of loop of Henle, transport mechanism is predominantly a passive phenomenon and permeability to water is less. Though the passive transport of sodium chloride occurs to some extent, active transport of sodium is almost nil. Thick ascending limb of loop of Henle, the fluid becomes more dilute, that is hypotonic, as sodium and chloride are taken out of the tubular fluid. The active transport system for sodium is well developed in this particular segment. Reabsorption of sodium from tubular fluid is dependent on carrier protein on the apical membrane that transport 1 sodium, 1 potassium and 2 chloride from the lumen into the cell. 1 sodium potassium 2 chloride co-transport membrane protein is also present in airways, epithelial cells of GI tract and salivary glands as well. From tubular cells, sodium is transported into interstitial fluid by sodium potassium pump, which is quite efficient in thick ascending limb of loop of Henle. Potassium from the cell diffuses back into the tubular fluid and also into interstitium passively through ROMK and other membrane potassium channels. Chloride that enters the cell co-transported with potassium into the interstitial fluid. Transepithelial transfer of these solutes creates a potential difference between tubular and interstitial fluid space, which drives the transfer of solutes, especially transport of sodium, potassium, magnesium and calcium via paracellular pathway. Thus, transport of solute occurs by both transcellular and paracellular route. However, the tubular epithelium of thick ascending limb of loop of Henle is almost impermeable to water. Therefore, solute transfer in this part of the tubule is not associated with transfer of water. Defect in thick ascending limb transport process causes Bartter syndrome. That is, the defect in the thick ascending limb transport process causes Batter syndrome. So this is how the reabsorption of the sodium occurs through sodium potassium 2 chloride channels in the thick ascending loop of Henle, and sometimes sodium, calcium, some amount of magnesium and potassium, they are transported paracellularly, and the potassium is taken back into the tubular lumen, sometimes through the ROMK potassium channels. Reabsorption of water. The loop of Henle reabsorbs approximately 15% of the filtered water, which occurs exclusively in the descending limb, the ascending limb being impermeable to it. The descending limb, except at its terminal portion, is highly permeable to water. In spite of active transport of solutes in thick ascending limb of loop of Henle, water is not reabsorbed. Both thin and thick portion of ascending limb of loop of Henle are almost impermeable to water. Furosemide is a loop diuretic which is going to act at this portion. Then functions of the distal convoluted tubule. Reabsorption of solutes and water. The distal convoluted tubule is essentially the extension of thick ascending loop of Henle. Hence, physiologically it is close to thick ascending loop. Therefore, the DCT is relatively impermeable to water, whereas solutes are transported. As solute is removed in excess of solvent, further dilution of the tubular fluid occurs in this part of the nephron. In the initial segment of the distal tubule, sodium chloride are reabsorbed from the tubular fluid into the tubular cells by sodium chloride transporter. The sodium is removed from tubular cells to interstitial space by sodium potassium pump located on the basolateral membrane, and chloride is removed by chloride channels. However, in spite of osmotic gradient across the tubular epithelium, water reabsorption is poor. Only about 5% of the filtered water is removed in DCT. Thus, dilution of tubular fluid that begins in the thick ascending limb of the loop continues in the DCT. Thiazide diuretics are going to act on this part of the DCT. Functions of collecting duct. Though collecting duct is considered as a distal part of nephrons, strictly speaking, it is not part of an individual nephron. However, as solute and water reabsorption continues in collecting duct system, physiologically it is essential component of a nephron. As collecting duct is the terminal part of the nephron, its ability to alter transport of solutes and water contributes considerably to final osmolality, composition, and volume of the urine. Most of the excreted potassium in urine comes from the collecting duct, as it secretes a significant quantity of this cation. Transport process in collecting duct are influenced by and fine-tuned by hormones such as aldosterone and ADH. Reabsorption of solutes in this part. Epithelium of collecting duct contains two types of cells, principal cells and intercalated cells. The principal cells reabsorb sodium and secrete potassium, whereas intercalated cells secrete either hydrogen or bicarbonate. Thus, principal cells contribute to potassium metabolism, and intercalated cells play an important role in acidification of urine. Intercalated cells also reabsorb bicarbonate and potassium. Principal cells are present abundantly in the epithelium of collecting duct. The reabsorption of sodium and secretion of potassium in the principal cells depends on sodium potassium pump in the basolateral membrane. This pump provides chemical gradient for movement of sodium from distal tubular fluid into the cells, and potassium is secreted from the blood via principal cells into the tubular fluid. Potassium secretion in the collecting duct is mainly occurs in the cortical part. In physiological condition, most of the potassium in urine comes from collecting duct secretion. Though in conditions of potassium excess, like high potassium diet, collecting duct secretes more potassium, and in potassium depletion it reabsorbs potassium. And potassium secretion is facilitated by aldosterone. Intercalated cells. These cells are scattered among the major cells of the collecting duct. On the luminal side, the membrane contains a sodium potassium ATPase that pumps hydrogen into the cell and potassium into the tubular fluid. In the cell, the hydrogen and bicarbonate are formed from dissociation of bicarbonic acid secreted into the tubular lumen and collecting duct. The bicarbonate is reabsorbed into the interstitial space. Thus, intercalated cells contribute to the acidification of the urine and conservation of the potassium. Okay. So this is how the bicarbonate, they are reabsorbed back into the basolateral membrane, and the H plus ions are secreted into the tubular fluid in exchange of potassium through the hydrogen potassium ion. Role of aldosterone. Sodium is reabsorbed from the tubular fluid by principal cells diffusion through sodium channels in the apical membrane of epithelial cells. These sodium channels are known as epithelial sodium channels, or also ENAC, that is epithelial sodium channels. Aldosterone facilitates genomic activity to produce SGK and other proteins that form epithelial sodium channels in the cells and their insertion into the luminal membrane. Thus, aldosterone increases the activity of epithelial sodium channels and increases reabsorption of the sodium. Sodium that enters in the cell is pumped out into the interstitial space by sodium potassium pump, the activity of which is also promoted by aldosterone. Thus, aldosterone acting on collecting duct increases sodium absorption and facilitates potassium excretion. Therefore, hypernatremia and hypokalemia are features of hyperaldosteronism, and potassium sparing diuretics inhibit sodium absorption in the collecting duct, and thus acts like a potassium sparing diuretic. This is the action of aldosterone, where it is going to act on HGK and other proteins. They increase the expression of epithelial sodium channels and also increases the sodium potassium ATPase, and which are going to help in the reabsorption of the sodium through the epithelial sodium channels. Reabsorption of water here. The collecting duct has two parts, cortical part and medullary part. The reabsorption of solute and water from the collecting duct mainly depends on the efficacy of ADH acting on it. In the cortical part, ADH increases the permeability of the collecting duct to water by inserting aquaporin to water channels, especially on the luminal membrane of the principal cells. ADH binds with V2 receptor on the basolateral cell membrane, whereas it activates adenylate cyclase to increase the production of cyclic AMP. Cyclic AMP activates protein kinase A, which promotes the insertion of vesicle containing aquaporin 2 on the luminal surface of the epithelial cells that increases water permeability. Cyclic AMP also increases gene transcription that increases formation of aquaporin 2. In the presence of ADH, water moves out of the hypotonic tubular fluid into the cells of the cortical collecting duct, and then from there into the interstitium of the cortex. This helps in reabsorption about 10% of the filtered water, and the tubular fluid becomes isotonic. The isotonic fluid then enters the medullary portion of the collecting duct. In the medullary part, the collecting duct osmolality of the interstitium is very high. The water is reabsorbed from tubular fluid into the hypotonic medullary interstitium by osmotic effect, which accounts for about 5% of the water reabsorption. This plays an important role in the countercurrent mechanism, and makes the urine concentrated. In human beings, in the presence of adequate ADH, osmolality of the urine may reach 1400 milliosmoles of water. In the absence of ADH, the cells in the collecting duct are relatively permeable to water. Therefore, urine becomes hypotonic in the absence of ADH, where osmolarity can be low as 30 milliosmoles per liter. However, the collecting duct is not totally impermeable to water, and therefore, even in complete absence of ADH, about 2% filtered water is reabsorbed. Coming to the tubular dysfunction, Liddle syndrome. This is a rare autosomal dominant disorder characterized by clinical features of similar to hyperaldosteronism, consisting of increased ECF volume, hypertension, hypokalemia, and metabolic alkalosis. However, aldosterone and renin levels in plasma is less. In this disease, the amyloid sensitive sodium channels become hyperactive, which results in absorption of inappropriately large quantities of sodium and increased potassium excretion from the tubular fluid. This is accompanied by reabsorption of large quantity of water. Thus, ECF expansion occurs that causes hypertension. Pseudohypoaldosteronism. There are two types of this, type 1 and type 2. Type 1, this is a rare autosomal disease which is just opposite to that of Liddle syndrome. The dominant form is due to the mutation of mineralocorticoid receptor gene, and the recessive form is due to the mutation of amyloid sensitive epithelial sodium channels. The disease is characterized by significant reduction in the ECF volume, and therefore condition is associated with hypotension. The decreased sodium channels and the decreased aldosterone activity in the tubule impair sodium reabsorption. Consequently, increased sodium excretion is accompanied by excretion of large amount of water. This causes the contraction of the ECF compartment and hypotension. In pseudohypoaldosteronism type 2, which is also called as Gordon syndrome, which is autosomal dominant disease, hyperkalemia, hypertension, and metabolic acidosis occur secondary to overactivity of thiazide sensitive sodium chloride cotransporter in the DCT. Gitelman syndrome is autosomal recessive disorder that has a similarity with the features of Bartter syndrome, like hypokalemia, metabolic alkalosis, salt wasting, elevated renin and aldosterone level, and normal BP. However, hypomagnesemia and hypocalciuria differentiated from Bartter syndrome. The disease occurred due to the mutation of thiazide-sensitive sodium chloride transporter, and decreased sodium absorption in DCT leads to volume depletion and hypokalemia. Loss of activity of thiazide-sensitive transporter increases the calcium reabsorption, causing hypocalciuria. So coming to the summary of tubular function, from the above discussion on tubular functions, we learned that nephron has functionally divided into three parts: proximal, middle, and distal segment. In the proximal nephron, the proximal tubule that reabsorbs about 67% of the solutes and water. Proximal nephron is non-selective and reabsorbs solute and solvent maximally. Thus, it greatly contributes to electrolyte and volume homeostasis. In proximal nephron, sodium and water reabsorption are closely coupled as epithelial water permeability is very high, and paracellular pathway of absorption of solute water is also well developed. In spite of small osmotic gradient across the epithelium, the quantity of water absorbed is large due to the greater permeability of the water in PCT. However, the tubular fluid is isoosmotic. The middle nephron loop of Henle constitutes the middle. In this segment, reabsorption of the solute in excess of water contributes to hyperosmolarity in medullary interstitium and makes the tubular fluid hypotonic. Thus, LOH plays important role in generating hyperosmolar interstitium that is essential for countercurrent mechanism of urine concentration. The distal nephron and the collecting duct, they are going to form the distal part of the nephron. Distal nephron is selective in reabsorption and absorbs smaller quantity of water and solutes. About 8% of the sodium and 18 to 20% of the water is reabsorbed in this part of the tubule. Sodium and water reabsorption are not coupled as water permeability is poor in distal portion. The tubular fluid is hypotonic. In spite of steep osmotic gradient across the epithelium, reabsorption of water is extremely poor in the absence of aldosterone and ADH. The paracellular route of transport is not well developed as the junctions between epithelial cells are really very tight. The distal nephron contributes to the concentration and acidification of the urine. So this diagram shows the important functions of the proximal convoluted tubule, where it helps in absorption of the water, sodium, potassium, glucose, amino acids, chloride, bicarbonate, urea, and also secretes hydrogen, ammonia, urea, and creatinine. Where the loop of Henle helps in absorption of the water, sodium, potassium, chloride, it secretes urea, creatinine. Whereas distal convoluted tubule, it is going to mainly the reabsorb water, sodium, chloride, and whereas collecting duct, it helps in reabsorption of water, sodium, urea, and it also secretes potassium, hydrogen, urea, depending on the situation. And this is the magnitude of reabsorption of water and various solutes expressed in percentage. Okay, and this is the glucose. Almost by the end of proximal tubule, almost all the glucose is reabsorbed, and the osmoles, the solutes and sodium, by the end of the collecting duct, the sodium and osmol solutes, they are going to be reabsorbed. The urea is going to increase in the middle nephron because it is secreted there. Then almost by the end of the nephron, only the 50% of the urea is reabsorbed. Coming to the regulation of ions and water reabsorption. Normally, the regulation of sodium chloride and water reabsorption. Normally, the total sodium is filtered is 2600 milliequivalents of in a glomerular per day, in glomerulus per day, and 99.4 is absorbed in the tubule. Of the filtered sodium, about 67% is reabsorbed in the PCT, 25% in loop of Henle, 5% in DCT, and 3% in collecting duct. The reabsorption of the sodium facilitates the reabsorption of many other solutes as we discussed and establishes the osmotic gradient for reabsorption of water. The total urinary sodium output ranges between 1 milliequivalents to 400 milliequivalents and average about 150 milliequivalents per day, depending on the dietary salt intake. Regulation of sodium chloride and water reabsorption, primarily the function of the various hormones acting on the kidneys. Neural factors, Starling forces, tubulo-glomerular feedback, and glomerular-tubular balance also play important role in regulation of sodium. The major hormone that influence sodium chloride and water reabsorption are angiotensin II, aldosterone, ADH, dopamine, glucocorticoid, urodilate, and atrial natriuretic peptide. Angiotensin II acts mainly on the proximal tubule of the kidney. It increases sodium chloride and water reabsorption and causes expansion of the ECF. It is secreted in the conditions of hypovolemia and hypotension. Captopril that inhibits angiotensin converting enzyme decreases the ECF volume and blood pressure. This prevents the formation of angiotensin I to angiotensin II. Therefore, decreased concentration of angiotensin II decreases sodium chloride and water reabsorption from proximal tubule and decreases vasoconstriction. This is used in the treatment of hypotension and in conditions of ECF volume expansion. Aldosterone stimulates the absorption of sodium chloride and water from collecting duct. It acts on sodium potassium pump and epithelial sodium channels. It facilitates potassium excretion in the tubular fluid. Thus, it increases the sodium concentration in the plasma and decreases potassium concentration. Among the important regulator of aldosterone secretion are hyperkalemia and angiotensin II. ADH is also important hormone for volume homeostasis. It is secreted from the posterior pituitary in response to hyperosmolarity of plasma and hypovolemia. It increases the permeability of the collecting duct to water. The dopamine inhibits sodium chloride and water reabsorption in the proximal tubule. Thus, it causes natriuresis and diuresis. Dopamine secretion increases in expansion of the ECF volume. Glucocorticoids have a mild mineralocorticoid activity. They increases water and sodium reabsorption. Urodelatin is a polypeptide containing 32 amino acids secreted by the distal tubule and collecting duct. It acts on the kidney tubules locally. It acts exclusively on the collecting duct to inhibit sodium chloride and water reabsorption from medullary part of the collecting duct. It's a potent natriuretic and diuretic hormone like that of ANP. However, recently the validity of this hormone has been questioned. Atyl nitriuretic peptide, it is hormone containing 28 amino acids secreted from the myocytes of atria of the heart. It acts on the distal tubule and collecting duct to inhibit sodium chloride and water reabsorption. Thus, it produces natriuresis and diuresis. It is secreted in response to ECF expansion and hypertension. Neural factors. Neural regulation is mainly through the sympathetic innervation of the kidney that controls water and sodium chloride reabsorption. Activation of sympathetic fiber results in increased sodium chloride and water reabsorption from the proximal tubule, thick ascending limb of loop of Henle, DT and CD. Actions are mediated via release of norepinephrine, epinephrine from the sympathetic nerve endings locally. The stalling forces regulate sodium chloride and water reabsorption across the proximal tubule. The peritubular capillary oncotic pressure and hydrostatic pressure in the interstitial space favor this process. The interstitial space oncotic pressure and peritubular capillary hydrostatic pressure oppose this process. When solids are reabsorbed from the tubular cells into the interstitial space, osmolarity of the interstitium increases. This favors the movement of water from tubular fluid into the interstitium via transcellular and paracellular pathway. Tubuloglomerular feedback mechanism. The signals originating from the renal tubules provide feedback for control of glomerular filtration, which is called as tubuloglomerular feedback. In this process, the rate of flow of and the concentration of sodium chloride in the tubular fluid of thick ascending loop of Henle and the initial part of the distal tubule provides feedback information to the glomerulus to control rate of infiltration. When the rate of flow through the tubule increases, the rate of filtration decreases, and when the rate of tubular flow decreases, the rate of filtration increases in the same nephron. Thus, the change in the glomerular filtration maintains constancy of the tubular load, that is, the constant volume of fluid is delivered to the tubule. The macula densa plays an important role where the macula densa provides signal to the afferent arteriole when tubular flow is high and concentration of sodium chloride in the tubular fluid becomes very high. The more quantity of sodium chloride enters the macula densa cells via sodium, potassium and 2-chloride transporter located in the apical membrane increases the sodium in the cell and increases the sodium potassium ATPase activity. Increased ATP hydrolysis results in the formation of more adenosine, and adenosine act on the A1 adenosine receptor to increase the release of calcium from macula densa that causes constriction of afferent arteriole. Thus, glomerular filtration is decreased. opposite mechanism operates when tubular flow decreases. It is also believed that similar mechanism operates in the control of renin secretion from JJ cells that influence glomerular function through the angiotensin II. And also, change in the diameter of afferent arteriole is mediated by secretion of chemicals like thromboxane A2 and prostaglandins locally. Alteration in glomerular filtration is also achieved by changing the surface area for filtration, which is brought by the constriction of or relaxation of the mesangial cells. Glomerular-tubular balance. Increase in the GFR increase the load of solutes, especially sodium in the tubular fluid. Thus, increase the reabsorption of the sodium from the proximal tubule. Normally, a constant fraction of filtered solute is reabsorbed. That means a constant percentage of tubular load is reabsorbed by the proximal tubule, and this is called as glomerular-tubular balance. So there are two possible mechanisms. One is change in the oncotic and hydrostatic pressures between peritubular capillaries and lateral intercellular space. This mechanism is more effective for sodium. Increase in the GFR increases the protein concentration in the blood that leaves the glomerular capillary. That is, the protein content becomes more in different arterioles. The efferent arterial blood enters the peritubular capillaries. Therefore, the oncotic pressure in the peritubular capillary increases, which causes increased sodium reabsorption from the tubular fluid. This is also accompanied by increased fluid movement in the interstitial space and then from there into the blood. Increased load in the tubule. With increased GFR, filtered load of sodium, glucose and amino acids on the tubule increases. Increased sodium and reabsorption associated with the increase in the glucose and amino acid reabsorption through the same carrier proteins. Thus, more solutes are reabsorbed. This is how the glomerular-tubular feedback is going to work, where there is increased GFR, the macula densa is going to sense more amount of the sodium, and that is going to increase the adenosine concentration, and that is going to cause the constriction of the afferent arterioles to decrease the GFR. Regulation of the potassium. Potassium is a primarily intracellular cation, and its gradient across the cell membrane is mainly determined by the electrical potential across the membrane. As the excitability of the nerves and the muscle depends on the electrical potential, precise control of potassium level is very essential, in which kidneys play important role. Normally 600 milliequivalents of potassium is filtered, and 99% is reabsorbed of that. The major absorption of the potassium takes place in the proximal tubule, which removes about 65%, and the loop of Henle removes 25 to 30%, and however 50% of 50 milliequivalents of potassium is secreted from the distal tubule, and thus net excretion of the potassium in urine is about 90 milliequivalents per day. The minor reabsorption and secretion potassium also takes place in collecting duct, especially by the intercalated cells. The rate of secretion of potassium by distal tubule cells is proportionate to the rate of flow of fluid through it. When the flow in the distal tubule is rapid, the secretion is more. This is because with the rapid flow, the potassium in the tubular fluid gets less opportunity to prevent potassium secretion. The distal tubule, usually sodium is reabsorbed and potassium is secreted. Though the transport of potassium is mainly passive, the movement of sodium from the tubular fluid decreases the potential difference across tubular cells. Thus, potassium movement into the tubular lumen is favored. The sodium is reabsorbed along with the secretion of H plus into the lumen. Therefore, a competition between H plus and potassium exists for exchange with sodium. When potassium in the body increases, H secretion decreases, and when H plus secretion increases, potassium secretion is inhibited. The potassium secretion is mainly regulated by aldosterone. So this is how the potassium is 65% is reabsorbed in the PCT, 20 to 30% reabsorbed in the thick ascending loop. It is 50 milliequivalents secreted, and some minor secretion and reabsorption occurs in the distal collecting duct, and thus the 90 milliequivalents of potassium is excreted per day. So this is in brief about the functions of loop of Henle, the distal convertible and collecting duct. Thank you.